Good morning and welcome to For the Love of Digital PCR 2024, an immersive virtual event dedicated to exploring the remarkable capabilities of digital PCR. We're thrilled to have all of you join us today. Our carefully curated program brings together a community of forward-thinking researchers, providing valuable insights and techniques to reimagine what is possible with digital PCR. By registering for this year-long event, you'll have exclusive access to ongoing content updates and insights from dPCR experts, empowering you to propel your own work forward. After today's session, we encourage you to explore our virtual environment. From our lobby, you can access main stage auditoriums no matter where you are in the world. Visit our poster hall to explore scientific posters showcasing the use of the QuantStudio Absolute Q dPCR system and auto-run dPCR suite across various applications. In our Education Center, you'll find relevant content including brochures, helpful videos, and links to additional resources. And finally, our virtual lab offers a sneak preview of how the Applied Biosystems family of genetic analysis tools can support a variety of research workflows. Now let's kick off today's event, which will be divided into two sections. In the first section, we'll hear from three invited guests who will discuss the impact of digital PCR on cancer research applications. In the second section, I'll be joined by two digital PCR technical specialists who will delve into the differences and similarities between qPCR and dPCR, as well as provide some tips and tricks to help get started with your own digital PCR experiments. Thank you again for your participation, and I'll now hand it over to Christy to introduce our first speaker.
Thank you, Christina. We are looking forward to an engaging event, and we encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many of your questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing the presentation. Now I have the pleasure of introducing Dr. Christopher Mason. Dr. Chris Mason is a professor of genomics, physiology, and biophysics at Weill Cornell Medicine and director of the World Quant Initiative for Quantitative Prediction. His laboratory creates and deploys new genomics technologies and predictive algorithms for medicine, which helps to ameliorate human disease and lays the foundation for long-term human survival on other planets. Dr. Mason's work spans over 300 peer-reviewed papers, 5 patents, 5 diagnostics tests, and the foundation of 10 biotechnology companies and 3 nonprofits. Dr. Mason also holds affiliate faculty appointments at the New York Genome Center, Yale Law School, and the Consortium for Space Genetics at Harvard Medical School. He is the author of The Next 500 Years, Engineering Life to Reach New Worlds. For more detailed speaker bio, please see the presenter tab at the top of your screen. Dr. Mason, welcome. You may now begin your presentation.
Thank you so much. Really, it's an honor to be here and a pleasure to join this group. And thanks for the invitation. And also make sure the sound's coming through loud and clear. Looks like it is. So great. And yeah, so I'm going to walk you through some of the work in our laboratory, which is focused on new ways to map minimum residual disease in sort of rare cells and rare alleles, and using digital PCR and sequencing and other methods to characterize cancer more broadly. So I'll kind of walk you through some of the recent work in our lab that spans a number of domains, and also places where we've been using Thermo Fisher. It really is kind of drivers for our research to map discoveries and even sometimes discover new things about Earth and space and all sorts of places. But today, focusing more on the clinical context, I'll jump right in. And so first we'll do a disclosure slide where, this is not an official endorsement of any particular product. You can see here some of this disclaimer. Also I've provided an honorarium as a disclosure for this talk. So what's exciting to think about, the world in which we live today is that, well, like I said, PopSeq, or population scale sequencing, has really become a global phenomenon, is really embedded into most large scale health systems, many countries, many statewide systems. It's being embedded into electronic medical records, or EMRs. We're seeing a really extraordinary time period in which we live, where the bedrock of germline information and even cancer information is being mapped with sequencing efforts. We're characterizing people at baseline, at relapse. We're getting all this information, and as a geneticist, it makes me very happy to see these sort of large-scale projects emerging. But when you do get diagnosed, of course, this is wonderful to think about all this baseline data. But when people do get diagnosed with cancer, there is always this challenge where what drives the emergence of the clone. You can see kind of here, this figure from a paper from Tim Lay and others. from a few years ago, but that we see this emergence in gray of a clone where something a cancer has emerged and is becoming a predominant clone. But after therapy, it changes, and you can get different clones, and you can have, essentially residual disease. And there's a lot of questions of how we can map this. And, some of the methods are for sequencing or digital PCR, which I'll get into today. But, there's a lot of questions you can see on this slide that we really are still teasing out, which includes what's the difference of the stem cells? How do they respond? How do we map MRD or minimum residual disease? What triggers the relapse? And we see heterogeneity at many layers, at cellular levels. We see it at genetic, epigenetic, protein levels for epitopes. And we know there's many, many layers of this heterogeneity that we can now see, even what are called epialleles, where you have, clonal outgrowth at the epigenetic level that you don't even see at the genetic level sometimes. So we and others have published in this work, and then it's, opens up a lot of questions that we still are teasing out in the clinical data. And in particular, one of the big questions is, what is the heterogeneity of cancer and MRD, and how do we measure that? And so, of course, you want to look for these, relapse clones at remission. Sometimes we see quadruple clones. You can see sometimes we see differences between clonal hematopoiesis, which I'll come back to a little bit at the end, and minimum residual disease, where sometimes CHIP, of course, may just be part of a passenger mutation that is just showing long-term risk for cardiovascular disease or cancer, but is not necessarily driving the disease. But also, how do we model this? And you can see there's just some plots of different ways we can see movement of different alleles between remission and relapse after diagnosis for different patients. And so, there are many, many ways in which, and many published papers. Actually, there's a great review paper by Rahul Satija from a couple of years ago that shows if you want to look at lineage changes across cells, even single cells, for example, there's now, a real large plethora of different methods to track, differences at epitopes, methylation, sequence, chromatin accessibility, and many ways to calculate what's called pseudotime, or this trajectory of these individual cells. And just to give you a preview of some of this, I want to demonstrate what we've been trying, I guess, in the lab as ways to tease out, these differences in cells. One of them, for example, is a lot of single cell RNA sequencing. Or in particular, if you look at what should normally happen, just to remind for you what it should occur, for example, for a normal lineage progression for lymphocytes, what happens in your bone marrow is you should see these hematopoietic stem cells. We see myeloid progenitor cells, these multipotent progenitor cells, and then lymphoid and common myeloid progenitors, or CMPs and CLPs. You can kind of see them on this penultimate layer for these different plots. And these are normal samples that we've profiled, where you can actually see, what's the fraction of cells that we pick up. And you can see we're picking these up with a CD34 positive, 38 negative sort for some of these cells. We're getting, you can see a lot of the expected precursor cell types. We're seeing that, in normal samples, they're either dominated by CLPs or MPPs, these different progenitor cells that, allow the bone marrow to become, everything it wants to be in terms of cell types. And this is, really what you should see normally. But of course, in AML samples, so for example, some ones we've profiled, we can see that this shifts, of course, right? So we're seeing a GMP population that's at relapse or refractory time points for some of our patients. We see it shift, right? So this normal progression of cell types that you should see in the bone marrow, of course, is shifted. And this is what leads to the disease, where we can start to see that, even some of the samples, for example, here, patient 9, this is transformed from MDS. It's refractory to standard therapy, standard induction therapy. And you can see, we're comparing this to peripheral blood. You can see even after therapy sometimes that you still see this shift down to a GMP phenotype. So these are some of the questions that you need to either address either with single cell methods or digital PCR, because it's really only subsets of the cellular populations that are sometimes subtly shifted, and they sometimes don't respond to therapies. You can kind of see here. There's another example here for MDS samples. This is an SRSF2 mutant. We see kind of the same thing. Instead of this normal progression, we see this shift across the samples that they're even shifting somewhat back to normal, but not quite. And they've changed from like a myeloid lineage to a lymphoid lineage, right? So even this progression of cell types coming down lineage specification, it changes, right? So this is what's fascinating to see, but really, it helps us drive new therapeutic directions for mapping it and then trying to target it in particular cases. And I'll come back to that later in the talk a little bit. But this just kind of visually shows you what's happening in the bone marrow. So to start to tease this out, before I'm going to start to look at some sequencing methods, also get into some digital PCR methods we've been using. But I'll start first with some of the sequencing methods. We've been looking at different splicing isoform mutant patients, so particularly MDS and AML patients where we see mutations in IDH or isocitrate dehydrogenase, and also spliceosome mutations in splicing relevant factors like SRSF2 SF3B1 and SRSF2. And you can see we've been looking at full-length long-read cDNA sequencing for some of these samples. And you can see here some of the normal cells we've profiled, as well as some of the IDH and the SRSF2 samples. This just gives you a quick QC of the number of cells we're looking at that pass QC. In particular for QC, for individual single cells, this is what we've been seeing for some of the insert sizes for our samples. Again, this is kind of a QC plot where you see the insert sizes generally hovering around 250 for its size. But, we do also see sometimes some longer, excuse me, we do see some longer inserts that do pop up for some of these batches of cells that we've looked at. But this is mostly QC. We do see often for the single cell data, the alignment's generally also high, getting 80 to 90% alignment, but we do see some tails of the distribution. Occasionally, this is, some individual cells have kind of lost what you can see, kind of this higher alignment rate. So we kind of clean these up and usually take the cells that have a generally higher alignment rate. So we see, this is one of the challenges of heterogeneity of processing these samples, that we can and do see differences in sort of the QC data that's coming out from these samples. Also, in particular, like what's the percentage of ribosomal RNA that we see, which is often an indicator of when we're seeing some stress cells or dying cells. And across different sequencing runs, you can see at the top, generally it's generally pretty low, but you do see, occasionally some spike ups. And I'll come back to this in a few slides as well, because we think in some cases this can even be a phenotype relative to healthy. We see this in disease, we see more of this. So even though you often use this as a cleaning metric, individual cell heterogeneity will sometimes show these differences that is actually a feature of the disease. It's actually part of what's possibly even driving the disease in some cases. And also mitochondrial DNA expression, also we try to keep this below 20% or even 10% if we can. And so now jumping into the data, that's kind of a lot of QC plots. Jumping into what do we see for these non-canonical splice junctions, this is using star2 alignments of individual cells. And you can see here, for example, the IDH and some of the splicing mutations, we see a lot more. So you can actually see, this really big jump up of what are the sort of, misdirection in splicing that, again, this is across all the cells. You can see the box plot here, the distribution across these cells. But it is itself a phenotype of the dysregulation of the disease, which is kind of fascinating to see. You can pick it up here in the single cell data. And so, of course, once we start to have these changes, we do see also lower percentage of coding bases in these IDH samples. So you can see here, normally you'd have a lot of bases getting, sequenced because it's cDNA, but you lose some of that in the IDH. You're getting all this dysregulation. You're getting almost read through for the RNA polymerase II that's probably going right through its normal regulatory landscape and just continuing to do transcription. You can see here. So they, see a lot. The healthy is way over here on the right, and we see the IDH way over on the left in terms of what we're seeing for the percentage of bases that are actually coding. And it doesn't really seem to be a function of batch. You can see down here on the right. So some other heterogeneity, we see more intronic reads in those IDH samples as well. So we're seeing this kind of spike up here for some heterogeneity that we've been picking up here in some of the samples, more intergenic bases as well. So things that you would expect to some degree. And this is, what we're seeing is real severe dysregulation in IDH1 and IDH2 mutant cases for our AML samples. So that's kind of a bit of background on heterogeneity, but then I want to talk a little bit about technical considerations of ways that we prep the samples and then ways we can do technical, variant detection using digital PCR actually. So first, when you prep a sample, as you can kind of tell, like a lot the QC plots we spend a lot of time on, because sometimes it's a phenotype, like the splicing dysregulation is QC, but then it becomes a phenotype for what we see in some of the mutants. But then also we see that we've been trying a range of different library prep kits with like NEB, Kappa, Covaris systems. Just a simple question of, I want to prep a sample for sequencing and what will I get out of it? And then if I'm going to do, for example, some PCR later or amplification, what will I pick up? So we've actually found in particular, again, there's a technical question, but it opens a lot of, I think, key doors into what happens in the tubes is, here's the GC content of the genome for H. influenza, E. coli, or a sort of palustris species. And you can see here, like the coverage is generally fine in the, mid-range of GC, but as you get to these really higher or low GC samples, you get variable coverage that is not based on, you want to be near zero if you can. So we've been playing with a number of different kits here to look at the coverage we see across samples. If you do human genomic DNA, it's a little bit more stable across most of the genome, but when you get to the outliers of the GC and you change your input amounts, that's where you'll start to see also some changes. It seems to be a little bit better with NEB. This is some collaborative work we've done with NEB on some of their newer FS kits, for example. And, you want to have good correlation between high and low input. We're seeing that doesn't seem to be too much of an issue. But this is where I think it's interesting is that we know that, if you look at the expected allele frequency versus observed, it mostly follows the line, but it's not perfect. For example, These are some of the horizon standards that we spiked into human DNA and then did digital PCR as well to detect some of the differences. And you can actually see, we can see them, but it's not always perfect, but it's mostly there. And so I think using some of these control methods and seeing how low can you go for the allele frequency, some of the most sensitive methods, again, this is done in partnership with New England Biolab, but we can see you get the digital PCR is often the way to get as low as possible for the expected VAF. And so that's kind of one other highlight of technical consideration. But another thing I want to compare to is early in the pandemic, to jump back to a COVID story, is we did, at one point we were doing a lot of PCR, LAMP, and RNA sequencing. Early in the pandemic, we were trying almost anything we could to a large degree. And this just showed some samples we looked at across subway samples. The hospital took basically all the first patients that were coming through New York Presbyterian. And with RNA sequencing, one of the first things we looked at first, for example, is what percentage of reads that were PCR positive for COVID versus PCR negative. You can see here, but you can also see it with RNA sequencing. You can see here that, using some of the PCR methods, we can pick it up, but then you can see there's high titer patients or medium or low, but these are all PCR positive. In some cases, the PCR is picking up, what are, barely detectable by RNA-seq, but definitely positive patients getting out to CT values of 32, 33, 30, even out to 35 sometimes. Interestingly, in the pandemic, I think you ought to remember this, that some people were putting their thresholds at CT of 40, which I thought was bananas, and then eventually got better, I think, in the pandemic, got down to 38, then 35, more to, I think, reasonable thresholds. But some of the digital PCR methods could definitely pick it up even out towards, 35, even 40 cycles. We could reliably see it out to, very low copy number, even maybe single copy numbers of virus. But when you look by RNA-seq over here on the right, you can see human RNA, bacterial RNA, of course, some other viruses. But a lot of the samples, you have to sequence really, really deep with RNA to actually pick it up. So this is one of the advantages of PCR, is that you can, of course, pick it up. That's why it was kind of the bedrock for most of the pandemic. We did compare it to LAMP, though, and also, I'll come back to that in a bit. In LAMP, for a little while, we couldn't get PCR reagents, so we were doing things a bit with LAMP, but PCR is still, I think, the gold standard for a good reason. And one of the things that we looked at first is, do we see co-infection, for example? Do we see at the top here, you see patients that don't have the virus, but based on PCR, versus low, medium, or high amounts of the virus versus no virus, but we picked up other viruses. So we can see here with PCR, we can pick up, other potential pathogens in these samples. And then we wanted to then compare basically how does the PCR compare to the RNA-seq. And PCR, again, is still, you can see is the gold standard. But if you look at the abundance we saw in the RNA-seq, you can pick it up. And if you look at detected based on PCR versus not, and then compare that to the amount of reads we saw with the RNA-seq, the RNA-seq, we can confirm that by PCR, but PCR is still, we're using as the gold standard here. So digital PCR is what we could actually pick up to actually see what was present. But there is a place, I think, for RNA-seq to serve as a good complement to what we see by PCR. And you can see here that the AURC is pretty good, sensitivity is pretty good, not as good as PCR, but is certainly, I think, interesting and compelling for basically just a complete shotgun RNA-seq with no optimization. So I think this does hold some promise. The other thing we liked about the RNA-seq is we do get a microbiome profile of the patient, so we could see which species were disrupted, basically in the nasopharyngeal swab, and the ones in red were the species that were disrupted, particularly in high titer patients. And then we also, because it's RNA sequencing, we get differentially expressed genes from the host, so we can see what genes are up or down regulated in orange and purple, respectively, and the volcano plot here showing statistical significance and the degree of change. And these pathways actually link to some things that make a lot of sense. So, for example, we saw interferon response upregulated. We saw gamma response, infection. We saw a lot of things that made perfect sense. But then other things that we did pick up, for example, that are interesting is like olfactory signaling completely turned off, or hemoglobin complex dysregulated. So we actually could start to see some of the phenotypes that we saw in patients, like the anosmia and the sense of loss of sense of smell, showing up in the RNA-seq data and the differentially expressed genes. So that was actually intriguing to see from the RNA-seq data. So the other thing we've seen, in some of the COVID patients, of course, is that we did some spatial mapping. We can see different kinds of cells were being picked up in the COVID patients. I can see here macrophages that are normally not present really at all in some of these lung samples were showing up in strong degrees in the COVID patients. So really disruption of who's there in cell types and what they're doing. And so we've published also an early COVID tissue atlas where we could see what happens, around the body, doing actually some RNA-seq, some digital PCR, some spatial transcriptomics across all these different samples. and published this work a couple years ago just to show like what's happening in the tissues. And one of the really striking, I'd even say creepy results that we got from from this paper in particular is we see, a lot of transcriptome damage, but also tissue identity loss in patients, which was really a peculiar phenotype. And what I mean by that is, if you look at what we see in these samples, you can see H&E staining is mostly similar structurally, but if you look at what's expressed here in actually these different tissues, if you look at the heart tissue, for example, you see some of the cells you'd expect to see, but in the patients with COVID high or COVID low measures by digital PCR and also by RNA-seq, you can see that the cardiomyocyte signature was gone. You can see down here versus in blue, the control samples, they still look like there's cardiomyocytes because you're in heart tissue, but it's lost essentially in these COVID patients. And so, the PCR, digital PCR defines sort of the thresholds. We're looking for high or low COVID patients, but in both cases, they really lost this, and we could then confirm that with RNA-seq. So really, I think kind of this haunting view of the cells are still there in the COVID patients, but they've lost sort of this regulation landscape of who they're supposed to be or what they're supposed to be doing. And we saw this across each different tissue. So across kidney, liver, and lung, we saw consistently a lot of, this loss of these sort of expected cell types that were showing up in the COVID patients. So another area that we work on in terms of characterizing patients, of course, is in cell-free DNA, cell-free RNA as well we look at. And so one other vignette where we've been using some of the PCR and digital PCR methods is looking at this question. So we have cell-free nucleic acids, which also, we do this for cancer patients. Obviously, there's a large competitive landscape of people doing, multi-tissue cell-free DNA sequencing for cancer profiling and MRD. But to remind everyone, you can get this from apoptosis, secretion, or necrosis. And then in the bloodstream, of course, there's many features of the DNA that you could use to characterize what's happening inside the body. And one very unique place where we use this actually was looking, for example, look at cell-free DNA and looking across all chromosomes. This is when Scott Kelly got back from space. So in the dotted lines was his year in space and before and after space flight. And one of the really striking things we've seen in him and in some other patients is a spike here you can see of this really mitochondrial DNA in cell-free DNA fraction. And so to confirm this, of course, we want to make sure, this was based on just shotgun sequencing of DNA, but then to confirm it using some digital PCR, we could actually see this again in copies per milliliter, published this work also a couple years ago. And we can start to see that the cell-free DNA and exosome profiles also showed this. So we can confirm this of what we kind of inferred based on the coverage maps of shotgun DNA sequencing. And indeed, in flight is really unusual phenotype where we're getting spikes of mitochondrial DNA we can show up. And so this we published a couple years ago, but it's actually some data that's coming out soon, probably in the next month or so, is, we publish, it's like a lot of case reports or publications. You see something interesting and you put it out there, but you of course would like to see it replicated in more cohorts and make sure it is validated. But what's exciting is this paper actually just recently accepted, now is coming out soon, that we also saw similar things. If you look at the count based on digital PCR, what we see for these mitochondrial spikes in plasma for cell-free DNA, this JAXA cohort, this Japanese astronauts, they also see this. So again, coming out soon, you'll see this paper. And if you look at the controls like mucin genes, we don't see this spike. This really seems to be particular to a stress response that you can pick up. That is, you pick it up with shotgun sequencing, but also with digital PCR, we can pick it up here, which I think is pretty cool. Also, just because it's validated some of our papers, so it makes me feel more confident that was really intriguing and seems to be a real consistent feature of spaceflight. And what we think this is actually you can, there is something that lymphocytes do called an interferogenic response, where they'll actually launch mitochondrial DNA webs in response to a stress, and basically as a way to kind of prime the immune system is one of the hypotheses of what we think is happening. And the reason I bring this up is it was for a few reasons. So some of these instances where we have large scale stress in the body, whether it's cancer patients, whether it's in clonal metapoiesis, or whether it's in astronauts, is it really begs the question of what's happening when we have inflammation occur in these big spikes, and there's a link to aging, and there's even a term called inflammaging, which if you've never done this, I recommend a Google image search that shows things like this, which I don't even know what that means, but it definitely doesn't look comfortable, right? So, but inflammaging is a concept, certainly in the field of clonal metapoiesis, where we know that as we age, of course, we all accumulate mutations, but we can see here that we want to understand these spikes of inflammation or things like spikes in mitochondrial DNA that you can pick up that are very subtle by PCR, or sometimes if you sequence deep enough. But we want to put this in the context of what we know also, generally about, some of the other, you can see it as a hot topic you can see on the lower right. But we want to put this in the context of what we know about, mutations, more broadly. So for example, here we know that everyone has mutations, and we did some work with Ross Levine a few years ago to see that even in controls, if you live long enough, you'll have TET2 mutations. And this is something that's been published also by work at Cornell, that you can maybe even, predict whether you're on the way towards getting leukemia 5, 10, even 15 years before the diagnosis. So the question is not, do you have mutations, but are they growing? Are the clones enlarging? Are you getting to a higher risk state for actually, getting a leukemia down the road? And the answer seems to be possibly yes for predictions. But, to do so, we really need to understand, the immune system, IL-6 spikes. And, if you sequence deep enough, you can find it. Like if you look for a VAF of 0.1% or 2%, you can see these two lines of, if you go very, very deep, you can pick it up. But this is something that's just really, impossible to do with sequencing. So a lot of this is done with digital PCR to get down to 1,000, 10,000, 100,000x coverage of very specific loci to pick up these rare alleles that are present in the blood. And what's interesting about clonal hematopoiesis is, if you sequence it, you find it. If you do PCR, you'll find it a little bit easier. But what's happening really inside the bone marrow is that the stem cells are encoding their own dominance, right? So progenitors here express anti-inflammatory factors to resist the inflammatory environment produced by their own progeny, which could eventually maybe clear them. And so in the literature, questions of how we address that has come up to say, well, maybe we could reduce it, such as like vitamin C in mice is one way that's been shown, again, only in mice, but shown as a way to reduce the levels of the mutated alleles. But when we look at clonal hematopoiesis actually in space, we've shown this, that actually, if you're in space, the number of the mutated alleles, the VAF, it decreased actually in space, which was interesting. And we confirmed this across multiple cell types, again, using some PCR methods here to confirm them and go to deeper profiling at low VAF. We could pick it up in sorted cell populations as well. But what's interesting is even though it went down in space, it did, eventually come back. You can see it. So it wasn't forever. It did come back, and we're now doing ongoing sort of clonal hematopoiesis tracking for these astronauts, as well as several others, as a sort of a long-term health assessment for these patients, as well as cancer patients. And actually, we often compare these patients to other ones, for example, at Sloan Kettering, where we can see here, what's the change in allele frequency? If you look over time, what this shows is, we only have a handful of astronauts, but if you compare them to untreated patients here in gray versus those that had external beam radiation therapy, and ideally, you can see the VAF, the change in the variant allele frequency could go down on the left or up on the right for DNMT3A or TET2. But generally, we can see that, what's happening in astronauts is not dramatically worse per se. This really just looks like aging generally, so inflammaging, maybe some inflammation driving it. But you can see here that this doesn't appear to be, at least from this cohort, dramatically out of line with what is expected just to see in general aging, and certainly not more than what we've seen for some patients who get radiation therapy and really dramatic changes in blue lines for the VAF that we see for these specific mutations in at least these two genes. So with that as a kind of a fun background, this is some work that we've recently, again, published this work a couple years ago. Fun fact about this, we did actually make a review figure, and then also if you zoom into the high-res PDF, you'll see that there's a picture of Baby Yoda that's hiding inside of that cell, which I think is cute. We'll put an Easter egg in your paper. You might as well do it when you're making a final review figure. And I would like to put a pitch in. I think it'd be great if we could actually get the QuantStudio working on the space station. I don't know, it is not yet planned by Thermo Fisher, but I'll put it a quick request to see if we get that working up there. Or eventually there's plans for a lunar base. I would love to also see a QuantStudio on the moon at some point. And I want to close in the last few minutes with a bit of a consideration of some data considerations, kind of a fun thought experiment to go back to the beginning question. If we're sequencing thousands and thousands or millions and millions of genomes around the world all the time, this gets into the range of big data. And some people are tired of hearing about big data. Some people get upset and they think of like Pulp Fiction's memes that say big data one more time. But the big data is coming from many places, not just population scale sequencing in countries, but a work that just came out this month is kind of a nice profile of ways where we're going out to detect like emerging pathogens, for example, sequencing all of our wastewater streams, environmental DNA, looking in subways, for example. And this is a great zoom in shot of some dual matrix Thermo Fisher tubes, which we use many of these per year. So you can see here kind of a quick screenshot from that video. But to go back to the data question, if we keep sequencing deeper and deeper, we might exceed astronomical levels of data and actually get towards genomical levels. There's actually a great paper that shows that a proper term is genomical to indicate large data, not astronomical, because genomical data is bigger and is probably getting bigger, getting beyond petabytes, exabytes, even zettabytes. And beyond zettabytes, you can get to a point of what's actually called a yottabyte, which is actually 1,000 zettabytes, or really it's 1 trillion terabytes. You'd have to fill the states of Delaware and Rhode Island with 1 million data centers just to store it. Actually, some people say yottabyte. I like to say yottabyte because I think it's a correct pronunciation. And just to give you a reminder, according to Yoda, you really only have 2 options, right? You can either do or do not. There's no try is not an option, right? So you have to do it. So let's just say we did that and decided to get 1 trillion terabytes. So it's about, these days, $50, $60 for 1 terabyte. And so yottabyte's 1 trillion terabytes. So you would need $60 a terabyte. That's like $60 trillion is what you'd need, which is more than the GDP of the entire world right now. Or it's a good chunk of it, I'd say, or more than any country. Certainly it gets about half of all the money in the world at this point. So, we certainly can't do we couldn't afford it today, really. Or maybe the world could altogether. But, it really begs the question of then, well, what do we keep and what do we track? And so in this regard, what we're spending a lot of time now is for MRD, for cancer, is doing very much what the internet is built for, which is finding cat and dog pictures, but also training a lot of the models on the rare VAFs that we're picking up in patients, finding what are the specific mutations we want to track more consistently. You probably don't want to keep every allele from every patient all the time, but learn when we get to the millions of patients or tens of millions of patients, which alleles are the most predictive, the most important to track, with digital PCR, with targeted capture sequencing, other methods. But these are the questions that, of course, eat up a lot of time just for image recognition. But I think in the coming years, when we get to these zettabytes and yottabytes of data, that we'll actually be able to tease out what's the most significant alleles to track that are the most predictive and across, of course, the most patients and help the most patients. So with that, I want to close and thank everyone in my lab that makes this possible. So I really couldn't be here today without their amazing and inspiring help for many of the projects I showed, as well as many others I didn't show today. But this is a really wonderful team I have the joy and pleasure to work with. Of course, I want to thank funding. This wouldn't be possible, of course, without funding from the NIH, the NCI, as well as World Content and Gates Foundation and the Pershing Foundation, also collaborators at NASA and AMH, and also other foundations you can see here. And then in closing, really want to thank again everyone in the lab, many collaborating teams. This is a partial list, but many great partners over the years who've made all this work possible. And I'm happy to stay and chat for a few questions. And thank you so much for your time.
Thank you, Dr. Mason, for that excellent presentation. Now, we have a few questions for you. I'd like to dive right in. Our first question, Dr. Mason, in your experience, do you feel that the trends of digital PCR assay development are heading towards highly customized or unique panels, or towards more specific outcome related to biomarkers?
It's a good question. So actually, I think we're seeing a lot more panels are being deployed, but I think they're going to see a lot more customization because it's easier now to swap out primers that work very well. And as you see different alleles emerge, for example, in different cancer patients, I showed examples where specific cells have mutations or different defects that you can now pick up. I think we'll see more customization in the longer run, though, because you could then have a battery of primers work well. And as soon as you see something interesting for a mutation, have an MRD panel that could even be, it's patient specific, but it's built off a catalog of primers that already work well. So you mix and match as needed the loci that you'd want to amplify, and then ideally track that for everyone. So, because companies like doing deep sequencing are trying this to some degree as well, where they're doing capture and then sequencing. But if you know the specific allele and you want to track it, you can obviously do it with some of the specific panels per patient.
Excellent. Thank you. And let's squeeze one more question in. With tumor heterogeneity and the landscape of mutations changing throughout the course of a disease, what strategies do you feel are most effective when selecting biomarkers and techniques for routine monitoring throughout the disease?
Yeah, I think digital PCR is the best way to get to very, very low VAF mutations that are present 1 in a million cells. But I think you can do it with sequencing if you have a panel and you sequence really, really deep. But if it generally at some point it just doesn't become cost effective, and so the digital PCR is the way to get to those really, really low, a single 1 or 2 copies of a mutation or of a molecule. And what's great is, though, I think you can get to the point where you're even at the cusp of biological relevance, right? So if something is present in 1 in a million or 1 in 100 million cells, that you may be able to see it, but does it matter? And so I think that's about where the point where digital PCR, you can get down to single copies potentially, but or even 2 copies or 3 copies. But even once you get to that level, you can see it. But what you want to be able to detect is it changing at all, right? So that's where you can say it was barely there, one or just a handful of molecules at one time point, but you look a year later or 6 months later, and you want to be able to really quantify, did something go from 3 molecules up to 20 or 10? And so that's where I think the digital PCR will help you in the very early stages of disease, or also in the more subtle response to relapse, for example, that for therapy. So yeah, that's but we'll look forward to seeing more of those kind of custom panels as the years come.
Dr. Mason, thank you so much for your time today and your contribution to digital PCR.
A pleasure. For the love of PCR, my pleasure, definitely.
Thank you. While we transition to our next speaker, we encourage you to watch this video about how the researchers at UC Irvine are leveraging digital PCR.
What we're doing has broad implications for impacting human health and disease. We really do feel that digital PCR is going to allow us to move forward with several really important fundamental questions. The focus of ongoing research in my lab is how the circadian clock is involved in biological processes and how disruption of circadian rhythms is implicated in cancer.
My current research focus is examining the types of mutations that lead to the etiology of several different B-cell malignancies like leukemias and lymphomas.
So the work in my lab focus on a protein called ApoBec2A and how this protein can promote RNA editing and create a point mutation at specific location in seconds inside a specific RNA.
Major issue that we had was really finding instrumentation that had the flexibility that we were looking for. Both myself, Remy, and Selma test out a variety of different platforms for digital PCR. And what really attracted us to the QuantStudio Absolute Q was both its flexibility and its sensitivity. It really, for us, hit that Goldilocks situation of the perfect amount of throughput for our needs. So for a lot of the different types of assays that we're building, we need to have a very sensitive assay that can detect low levels of mutations. And it also allowed the flexibility of looking at multiple sites through multiplexing.
Oftentimes, the amount of material that you get from patient samples is extremely limited. The QuantStudio Absolute Q allows us to get down to really low levels of material that's required for effective analysis, whether it's copy number analysis or point mutation analysis. This is one of the platforms that allowed us to go down to 1 nanogram of genomic DNA as a method for quantification of changes in the genome. This allows us to power up our assays to get the maximum amount of information that we can get from a small amount of genetic material.
This method is extremely fast and easy to use. It's a one-step process.
I don't think that there is a platform out there that's easier to use than this one. Have an instrument where you can just create a general PCR product, load it up, just put it onto a plate, put it into the machine and forget about it, has been great because it limits the amount of hands-on time.
The data quality that we've been getting from the QuantStudio Absolute Q has been absolutely fantastic. That data has been extremely reproducible in our hands. The software has been user-friendly. We've been able to basically just select copy number changes and the calculations are done automatically by the software. My lab has been really very, very happy with working with support through Thermo Fisher Scientific on this. We consider them really wonderful colleagues in terms of helping us to establish some of these assays in the lab. I really feel that this technology has the ability to revolutionize cancer-based research. We're able to take our science, not just from the lab, but translate our findings into the clinic. And without that level of sensitivity and reproducibility, that would not be possible.
Welcome back. To continue our session, I would like to welcome our next speaker. It is my pleasure to introduce Dr. Sebastian Dintner. Dr. Dintner currently serves as the head of molecular pathology at the Institute for Pathology and Molecular Diagnostics at University Hospital Augsburg in Augsburg, Germany. Today, Dr. Dintner will speak about multiple digital PCR approaches in molecular pathology. Again, as a reminder, we encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer as many of your questions as we have time for at the end of his presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing the presentation. Dr. Dintner, welcome. You may now begin your presentation.
Thank you for your kind introduction and welcome to the virtual event dedicated to exploring the capabilities of digital PCR. Today I will give some insights to the versatile approaches of digital PCR at our lab in Augsburg, Germany. My name is Sebastian Dintner. I'm responsible for the molecular diagnostics at the Institute for Pathology at the University Hospital in Augsburg. For short notice, the disclaimer of Thermo Fisher. The University Hospital is located in the southeast, close to Munich. It is one of the biggest hospitals in Germany, which provides maximum care for a region of about 2 million residents. And since 2019, the hospital is transforming into a university hospital with big construction measures and of the planned medical campus. Analog to that, we developed, equipped our diagnostics spectrum in the molecular pathology to compensate the evolving molecular diversity in tumors. Until the beginning of 2018, we applied genomic profiling with one capillary sequencing system, which was displaced by several modern analysis platforms in the last years. The need of comprehensive but also targeted genomic profiling is significantly increasing over the last years. Since there are increasing numbers of biomarkers shown here on the left side, has to be analyzed in personalized cancer care in the last 2 years, comprehensive genomic profiling, which allows us for the simultaneous analysis of a wide array of genomic alterations in cancer samples. It's a powerful tool to meet the need of modern cancer care. And at the same time, here shown and depicted on the right side, the COVID pandemic included increased awareness of the potential for zoonosis and emerging infectious disease that can affect human health. Although emergent viruses are currently in the spotlight, we must not forget the ongoing toll of morbidity and mortality owing to antimicrobial resistance in bacterial pathogens. With this diagnostic spectrum, pathology finds itself precisely in the area of potential competition, and it's looking for appropriate analysis methods for this diagnostic challenge. Comprehensive analysis using NGS technologies is now widespread, but the digital PCR offers several advantages and complements the NGS approaches very well. It has big advantages in absolute quantification without standard curve, high sensitivity, even at lower target conditions, it has very short turnaround times and is very cost effective. This makes dPCR a very good alternative in terms of liquid biopsy approaches, copy number analysis, pathogen detection, and verification of NGS results and gene expression analysis. We at our institute have, in addition to the classical PCR assays and fluorescence in situ hybridization assays, our institute focuses on the diagnosis of hematological neoplasms and solid tumor diseases. These are examined using various target panel solutions and supplemented with comprehensive analysis, including whole exome sequencing. We have also implemented a large number of pathogen detections and detection methods in routine diagnostic, as shown here in the slide. For the targeted detection of mutations, we have been working with the 3D PCR system from Thermo Fisher since 2017. The system had great advantages with its singleplex and chip-based system, but was somewhat complex due to its hands-on tasks and was also not as robust in use. The follow-up system, the Absolute Q, is in our lab since last year and impresses with its multiplex analysis and significantly improved user-friendliness and local data processing approaches. Thanks to the automated loading of maps, so the microarray plates, up to 16 analysis can be carried out very quickly in parallel. This is reflected above all the homogeneous loading of the chip and the quality results shown here on the right side. In the next few slides, I will show you the advantages of the Absolute Q system using examples of dPCR applications in our laboratory. First, dPCR in pathogen detection. Successful pathogen detection on tissue sections presents us in pathology with major challenges. Pathogen structures are already very difficult to detect on tissue sections, in this case a gram staining. It doesn't get easier at different magnifications either. So especially when you realize that pathogens cells are clearly in the minority compared to human cells and have a much smaller genome. We need very sensitive detection methods in order to be able to provide appropriate evidence. I will show you here an approach for Mycobacteria tuberculosis detection. So far, we have detected tuberculosis with a very robust lab development test PCR analysis for a purpose of specific gene. For this purpose, a specific gene coding for a heat shock protein is amplified and then subtyped using a restriction fragment length polymorphism. This can clearly differentiate between tuberculosis complexes and mycobacteria other than tuberculosis, as can be seen here in the gel images on the left side. This processing takes approximately 2 days. With the Absolute Q system, we can detect an Mycobacterium tuberculosis complex very quickly in just a few hours using the TaqMan microbe detection assay. As described above, this assay is based on a specific detection using a thumb probe. As shown in the figure on the right, we are able to distinguish very well between TBC positive and negative cases with the system and successfully set up an open AQA scheme for quality assurance in Germany. Second example approach is dPCR in monitoring. Since 2019, we have been working very closely with the Department of Hematology and analyzing almost 1,000 initial diagnosis and follow-up samples per year for molecular risk stratification of hematological neoplasms. This is done particularly in the initial diagnosis, but also over time with a myeloid panel solution on the GeneXus platform by Thermo Fisher for DNA and RNA changes. Patients should be tested very regularly to rule out minimal residual disease. This requires very sensitive methods in order to be able to detect even low frequencies of mutations. In this example, dPCR comes into play as a monitoring method. Here you can see the course of an AML patient which initially shows multiple changes in RAS, NPM1, NF1, ZRS2, and JAK2. The JAK2 clone expands over time and is therapeutically contained accordingly. In this first phase of disease, the progression was monitored using NGS. Then sequentially, the JAK2 variant was monitored by dPCR in order to be able to detect an early remission of the AML. Here, the counts of the individual points in time enable a limit of detection of around 0.1% of allele frequency. The previous 3D system worked very well here and is being replaced by Absolute Q with consistent quality, shown here in this one example. This works just as well for other targeted questions such as MPL, MyD88, BRAF, or the detection of the Philadelphia chromosome, BCA-ABL1. We can achieve a limit of detections of 0.08 in a very robust and reproducible manner, as shown here in a dilution series of JAK2. Third example for approach in liquid biopsy. In this last example, the multiplexing option on the new Absolute Q system comes into play. Last year, LSS Strand, a new generation of selective estrogen degraders, received an EMA approval for use in advanced breast cancer following endocrine therapy. Mutations in the ligand binding domain of the estrogen receptor are responsible for this resistance mechanism. For appropriate therapy, ASR1 mutations must be detected in the patient's blood plasma. For this purpose, together with Thermo Fisher, we designed the TaqMan probes for a multiplex assay that enable the parallel detection of 8 ASR1 hotspot changes. For this purpose, wild-type alleles and mutant alleles were marked with VAM and VIC and AB sci-fi and applied to a quadruplex analysis. An ASR1 mutation analysis can therefore be carried out using only four reactions. By participating in a comparative study, we were able to test the assay design on 14 artificial plasma samples. On the left side, we see the wild-type events in blue in the WIC channel and the mutated events in green in the WIC channel. On the right side, an example of the known scatter plot view is shown, which clearly differentiate the wild-type population from mutant population. Here also the representation of the AB sci-fi signals, which also enables a very sensitive and specific analysis of around 1% of allele frequency out of plasma samples. The dPCR method shows a 95% agreement with the results from NGS technologies, here compared to the results of the Genexus pipeline. The method is very sensitive and specific and is therefore suitable for use in routine setting. For this reason, we started to apply this assay in a routine setting. Therefore, I show you here the results of the first 20 liquid analysis to detect ESR ESR1 variants in the blood plasma. To do this, we are collecting 2 times 10 milliliter PAX gene or EDTR blood samples and isolate CCF DNA from it using a Maxwell protocol. The samples are then placed in the previous described ESR1 assay. And on average, we get 0.3 nanogram per microliter of CCF DNA from approximately 8 milliliter of plasma. This enables the detection of almost 1,000 events per mutation. In a few cases, it's significantly more, so around 3,000 to 4,000. Basically, we reliably achieve a detection limit of 1% allele frequency for all ESR1 hotspot changes. And the current prevalence of ESR1 mutated sample is around 60% in our collection. With that, I'm coming to a summary and to the end of my talk. So the Absolute Q system offers the possibility to monitor and identify changes with a high sensitivity in the course. So dPCR achieves a high sensitivity up to 0.1%. This has big advantages in monitoring rare mutations and approaches in liquid biopsy. And the absolute quantification without the need of reference standards is useful for applications like microbial detection. So dPCR is a fast and robust option for us to verify rare mutations identified by NGS. And the ability of multiplexing with the Absolute Q system offers a lot more applications in monitoring and identifying multiple rare mutations. These applications demonstrate the high versatility of dPCR in clinics for us. And with that, I'm at the end of my talk. Thank you for your attention, and I'm looking forward to an interesting discussion.
Dr. Dintner, thank you so much for your excellent presentation. So now it is time for our live Q&A portion of our webinar, and we have some great questions already coming in. So Dr. Dintner, I'm going to start with this very first question, and let's dive in right here. Where do you think are the benefits of using digital PCR together with NGS when doing oncology research?
So as I mentioned, one point is a benefit for using digital PCR is for monitoring. So when you identify certain biomarkers, you can follow them easily and very fast by digital PCR, very targeted. And another advantage of the system is when you identify a rare mutation which is not known, or you're not sure if it's because it's of low abundance with a low allele frequency, you can verify this variant very fast by this technology.
Very good. Now, Dr. Dintner, what's the lowest input you can go in terms of DNA quantity, and do you use the digital pooling?
So for liquid biopsy assays, it's quite important because, as I mentioned, the average concentration of CCF DNA is very low. So we have only 0.3 nanograms in average per microliter. So we try to add the maximum volume to the Absolute Q system. This is around 6 microliter per reaction. And this is, kind of the lowest concentration we add. For this reason, we running the system in technical replicates, and therefore we pulling this events later on by the analysis part. Yeah. So this is kind of an approach we regularly using. Yeah.
Very good. Thank you. And it looks like we have time for 2 more questions. What, in your opinion, would be the most informative targets for a multiplex ESR1 assay?
So targets in terms of biomarkers we would like to identify.
Very good. And our final
Yeah,
question.
yeah. So we choose the most abundant hotspots. So they're a bit more. I showed it on the slide. There are a bit more variants described in literature, but we choose the variants with the highest prevalence, and we are also thinking about to increase the numbers of hotspots on the Absolute Q system, but right now we are fine with the 8 hotspots we choose. Yeah.
Very good. And our final question, how do you select mutations for multiplex digital PCR assays?
So we combined different hotspots. So there are multiple base pair changes on one side, and we try to combine these hotspot with other hotspots. So, for example, at the locus 380, we try to combine different hotspots in one multiplex assay to have a nice signal differentiation between them. Yeah.
Dr. Dintner, thank you very much for your time today. And I do want to thank our attendees also for their engaging questions.
Thank you very much.
While we transition to our next speaker, we encourage you to watch this video about how the researchers at Hospital Puerta de Hierro are leveraging digital PCR.
So my name is Atocha Romero. I work at Hospital Puerta de Hierro in Madrid, Spain. Basically what we do is to evaluate the clinical utility of liquid biopsy for the management of lung cancer patients. We do non-invasive biomarker testing and we monitor cancer patients through liquid biopsy. Lung cancer is a devastating disease. It's a very deadly disease. Most of the people that are diagnosed with lung cancer patients die. And the only way to reverse this is to diagnose cancer in an early stage where cancer can be cured. So Applied Biosystem Solutions have technology that we are very interested on. So technology with a very low limit of detections, so for example, digital PCR, which we think will help us significantly to improve non-invasive biomarker testing or tumor response to treatment monitoring. We were one of the first users of the QuantStudio 3D digital PCR, and that was really helpful for us because we could analyze in a non-invasive way somatic mutations in lung cancer patients. Digital PCR is very useful to monitor disease, and monitoring the mutations when the patient has, or the tumor of the patient has a druggable mutation, monitor that mutation over the course of treatment is very helpful because together with the imaging information permits a narrow management of the patients. So my dream is that liquid biopsy can really help in the screening of cancer. So I really think that the development of radiomics and the combination of radiomics information and liquid biopsy will help in that, and hopefully we can offer an efficient cancer screening that can be carried out in the public health systems with an effective screening that will reduce substantially the mortality of lung cancer.
Now I have the pleasure of introducing our next speaker, Dr. Yanan Kuang from Dana-Farber Cancer Institute. Dr. Kuang received her PhD degree from University of Rochester and did her postdoctoral training at Dana-Farber Cancer Institute. Dr. Kuang's current research focuses on plasma-based biomarker analysis and clinical assay development. Prior to joining the Belfer Center, she was a scientific lead and research project manager at Variogenics, working on generating modified thermostable polymerase for customized nucleotides, and designing a high-throughput SNP identification platform. Today, she's going to be speaking about her work developing digital PCR assays for non-small cell lung cancer markers. Welcome, Dr. Kuang. You may now begin your presentation.
Thank you, Christy, for your kind introduction. Hello, everyone. I'm honored to be here to share with you our updates on using Absolute Q digital PCR platform to quantify plasma genotyping. In the past 10 years or so, our laboratory has been very interested in studying non-small cell lung cancer. This is what we know about the disease in year 2022. There are two categories in non-small cell lung cancer. The driver-negative category, who have different expression levels of PD-1. The patients all go through immunotherapy. The driver-positive category have the hallmark KRAS or EGFR mutations, as well as other missense and fusion mutations. For many of these mutations, they are drugs that improve patients' lives. Why do we want to study blood? Why are we interested in blood liquid biopsy? We truly believe by studying blood, we can take a peek into solid tumor biology. Blood biopsy may capture tumor heterogeneity. It is minimally invasive. It enables serial monitoring of cell-free or cfDNA kinetics and emergence of resistance. They complement traditional biopsies, which often are inadequate and insufficient for molecular analysis in 20 to 30% of clinical cases. This research from our group was published in 2016, JAMA Oncology. We conducted a prospective validation across 180 non-small cell lung cancer patients whose tumor was genotyped. They either carry an EGFR or KRAS mutation. We custom designed droplet digital or dPCR assays for the mutations. Results are shown to the right. Mutation burden is plotted in the units of mutant copies per mil of plasma. As you can see, the range was from 10 to 100,000 mutant copies per mil of plasma, over 5 orders of magnitude, showing how dynamic these assays are. Also, when we detected one type of driver mutation, we did not detect the other type of driver mutation in the same sample. It confirmed that the driver mutations are mutually exclusive and suggested that these driver mutation assays are 100% specific. Here, I want to draw your attention to EGFR T790M assay for an EGFR resistant mutation, showing in the second panel to the right. There were a few blood samples which were T790M positive. However, their corresponding tumor samples were T790M negative. The presence of false positive drove the specificity of T790M assay down to 63%. It suggests the heterogeneity of the tumor. In the same paper, we also reported a case study. This patient was in their early 80s, whose disease progressed on first-generation EGFR TKI erlotinib. On the same day, 2 assays were ordered, a plasma ddPCR assay, which resulted a T790M positive the next day, and a repeat biopsy, which made the physician wait for another 24 days. When the T790M positive was confirmed by tissue assay, osimertinib, a third-generation EGFR TKI, was initiated. Luckily, the patient responded well, and his tumor shrunk in size. Currently, in the setting of acquired resistance to EGFR TKI, FDA recommends plasma assay for T790M and sensitizing mutations. If T790M is positive in blood, the patient can skip biopsy and move on to third-generation EGFR TKI treatment. If T790M is negative, we still have to perform biopsy and use FDA-approved FFPE assay to test T790M. If T790M is positive, the patient proceeds to third-generation EGFR TKI treatment. If T790M is negative, the patient goes under chemotherapy. For our validated ddPCR assays for EGFR and KRAS, we can use them to monitor disease progression in advanced stage non-small cell lung cancer patients in longitudinal fashion. A 2014 CCR paper demonstrated such monitoring in 2 EGFR deletion 19 mutant patients. Upon treatment with first-generation EGFR TKI, driver mutation here showing in solid black dropped to 0, but with time bounced back in a classic V shape. The T790M resistant mutation in dotted line also emerged along with a reemergence of driver mutation with time. From these 2 examples, we can see that cfDNA shed can be variable in different samples. We can detect the cfDNA shed quantitatively over time. It holds true for both EGFR and KRAS gene detection, as well as genes in our other projects. Our lab at the Belfer Center also serves as a reference laboratory to do orthogonal clinical performance validation to compare ddPCR with new NGS technology such as Guardant Health. We have done ddPCR analysis with non-small cell lung cancer mutation assays. In addition, we have BRAF, ESR1 assays for other disease such as breast cancer. Between the 2 platforms, there's a nice concordance rate of above 95% in 221 patients. Especially for those with an AF greater than 0.5%, concordance rate is 99%. Only when AF drops to below 0.5%, the concordance decreased to 64%. When AF value is greater than 5%, the rate is as high as 99.1%. Now, when we zoom in to see how EGFR assays did for these 2 technologies, deletion 19 to the left, LA58R, and T790M to the right, the concordance rate is 96% for deletion 19, 100% for LA58R, and 96% for T790M. We truly believe that ddPCR can serve as a benchmark assay for broad targeted NGS assays. I have discussed EGFR assays extensively. Now I want everyone to take a close look at KRAS mutations. In this table, I listed major KRAS mutations in non-small cell lung cancer, PDAC, and colorectal cancer. For lung disease, 25% of mutations are KRAS mutation. Out of KRAS mutations, 85% of them are from G12 missense mutation. The most common G12X mutations in non-small cell lung cancer are G12C, for which 2 drugs have been approved by FDA. For G12D and G12V mutations, there are drugs already in the pipeline. Hence, it is of great interest for us to bin the patients to find their specific genotypes. Previously, we have worked out a collection of dPCR assays for KRAS. Here, we want to design a multiplex KRAS assay which can identify the most common and most therapy-relevant G12 mutations in non-small cell lung cancer: G12C, G12D, and G12V. In the meantime, also identify other G12X mutations using a drop-off probe. Since there are 4 channels we can use to probe these mutations, we assigned each channel to one common G12 mutation. The lineup and the colors are listed to the left, and sequences are shown in the right panel. The reference probe downstream of G12 position gives us the ability to differentiate the wild-type population from the missense G12X population. Using genomic DNA extracted from cell lines that harbor either wild-type or mutant KRAS mutations, we could guide mutation calls for our one-part KRAS assay. We have summarized the visual calls in 5 categories shown here. In the G12 wild-type sample to the left, there are equal copies of wild-type and reference counts. In a G12C sample, there's a high FAM cluster in addition to wild-type cluster in green. Next, a G12D or G12V sample presents either a specific RB or Cy5 cluster. Lastly, to the right, using our drop-off probe, we have a lower FAM cluster in addition to the green wild-type cluster. For those samples you don't have RB and Cy5 signal, which tells you it's not a wild-type, but belongs to a G12X genotype, which is non-G12C, non-G12D, and non-G12V. To complete our basket of assays, we try to convert our ddPCR assays for EGFR mutations onto digital PCR platform. Kit 1 is for EGFR driver mutations, which consists of the deletion drop-off assay and the missense LA5AR assay. Kit 2 is for EGFR resistant mutations on exon 20, the T790M, and 2 versions of C797S. The conditions of the assays are listed in this slide. In the next 2 slides, I'm going to describe the mutation calls for EGFR assays. This slide is for EGFR kit 1. There are 3 categories of calls for this kit. To the far left, there's the EGFR wild type, where you have 1 green cluster in FAM-Vic plot, in Cy5 alone cluster in RB Cy5 plot. A deletion 19 mutant presents itself with a Vic-positive, FAM-negative cluster in the middle panel. In the right panel, LA5AR mutant presents an RB cluster shown in purple. These are the results for EGFR kit 2 mutation call. Wild-type calls are shown on the far left. They are simple clusters. To the right, we have T790M alone with a purple FAM cluster in the absence of C797S. In the middle 2 panels, there we show 2 different versions of C797S in the presence of T790M. C797 never present themselves without T790M. After we established these digital PCR assays, we moved on to use 48 clinical samples extracted from plasma. The patients have previously been confirmed with EGFR or KRAS mutations in their tumor. We performed digital PCR as well as our established ddPCR assays and compare results. To the left, the AF percentage measured by DD and dPCR nicely correlated with each other. In the middle panel, we have the zoom-in version of the graph from the lower left register. In the table to the right, we show that the concordance rate is over 91%, or 42 out of 46 samples. To summarize, we have translated our EGFR and KRAS ddPCR assays to Absolute Q digital PCR platform. Absolute Q's multi-channel format allows us to consolidate EGFR assays to 2 kits and KRAS assays to 1 well kit. EGFR and KRAS Absolute Q digital PCR assays were validated using cfDNA extracted from plasma samples of 48 advanced non-small cell lung cancer patients with tumor-confirmed EGFR L858R, exon 19 deletion, T790M, and KRAS G12X mutations. EGFR and KRAS Absolute Q digital PCR assays have been shown to be as sensitive and specific as our validated ddPCR assays on Bio-Rad platform. We are in the process of clinically piloting our validated rapid genotyping approach in patients and quantify turnaround time advantage of multiplex genotyping in a cohort of non-small cell lung cancer patients undergoing standard of care tumor genotyping. Finally, my big thanks to patients who participated in our studies, to doctors, our groups at Dana-Farber, Drs. Powerlitz and Yanan, and our funding sources. And I want to thank everyone for your attendance. Now I can entertain questions.
Thank you, Dr. Kuang, for your informative presentation. It is now time for our Q&A session with you, and I have a few questions, so I'd like to start with this one. So Dr. Kuang, since the amount of circulating cell-free DNA that is shed over time can be variable from patient to patient, what have you seen through your research is the best indicator for recurrence using these biomarker-based assays? And is it overall quantity or the presence of specific mutations?
Yeah, in our initial studies, we actually have measured overall quantity of cfDNA, and our results found that overall quantity does not matter. What it matters is specific mutation burden.
Very good. Thank you. Our next question. How do you select mutations for multiplex panels?
So in the case of KRAS, we select the most common mutations in particular disease, and also whether they are therapy relevant. And it's the same with EGFR mutations as well.
Thank you. And I have one more question for you. Do you see an advantage in pairing next generation sequencing and digital PCR methods together for cancer research applications?
Definitely. So as I described briefly in my presentation, that we have, so the NGS and digital PCR give different answers. So NGS would provide mutations information across the whole panel of sometimes several hundred genes, right? So digital PCR, however, give you detailed information on targeted assays. So these are not screened, but specific, allele-specific assays. So they complement one another.
Thank you, Dr. Kuang.
You're very welcome.
While we transition to our next speaker, we encourage you to watch this video about how the researchers at University of Wisconsin-Madison are leveraging digital PCR.
Digital PCR, it's something that we've been wanting to do for quite some time.
We really focus on genome editing here in the lab, and if you're going to do genome editing, you want to have a lot of faith in the actual edit you made. So when we hand samples back to our users, we want to make sure that we're confident that's exactly the change that we think it is. And digital PCR is a key aspect of that quality assurance, and that's what drew us to the Absolute Q in the first place.
It's very simple to use, not many steps in handling. It's just load up the cartridge and go. I like the optimization abilities of the Absolute Q. You have a lot of difficult areas of the genome to probe. Customizable ability of the Absolute Q is extremely important for analyzing those regions.
The Absolute Q can multiplex and capture different channels at once. We can now gather data either on multiple control channels or measure multiple experimental samples.
The software is super user-friendly. Anything that you think you can get out of a single PCR reaction, the Absolute Q software can guide you through to that.
So we've been using porcine models to study human disease and cancers. We made a P53 mutant pig model, and the Absolute Q allows us to validate and make sure that the pigs that we made in each individual offspring have exactly the edit that we expect them to.
One thing I'm really excited about with the Absolute Q is I'm able to get reliable CNVs of up to a KB amplicon or a kilobase amplicon, and that's not something that I expected out of this instrument, and I'm excited to try to push that boundary a little further, see what more I can do with it.
It's kind of just the beginning of an era. There's a whole bunch more that we have planned in store, so we're pretty excited about it.
Thank you very much to Christy and our speakers, Dr. Mason, Dr. Dintner, and Dr. Kuang, for this first section of today's event. We hope you enjoyed these insightful presentations. Now we'll move on to the second educational section, where we will explore the differences and similarities between qPCR and dPCR, and receive valuable tips and tricks from our digital PCR technical specialists. Please feel free to continue to submit questions to the Q&A, and we'll follow up with these questions post-event. Our first speaker is Dr. Marcia Slater. Marcia is a senior technical specialist for digital PCR at Thermo Fisher Scientific. She has been at Thermo Fisher Scientific for 27 years, and over those years, she's held several technical roles with Applied Biosystems, Life Technologies, and Thermo Fisher Scientific. Her passion is PCR, especially qPCR and dPCR. Prior to joining Applied Biosystems, she was a research scientist at the Sharon Clow Research Institute, and she holds a BS from Penn State and an MS from Rutgers. Thank you, Marcia, and welcome.
All right, well, thank you so much. I'm really excited to be here and talk about my favorite things, which are anything having to do with PCR. With real-time PCR and digital PCR, the most common question I get is, which one should I use? What are these things, and how do I know which is the right one for my project? So let's start out with just a little bit of background, because there might be some folks here who haven't used one of these or the other. So I'll start with real-time PCR, and you're seeing a little clip of some real-time PCR data in front of you. This is showing the increasing of fluorescence versus the cycles of PCR. So with real-time PCR, we're capturing all of this data throughout the entire course of the thermal cycling. And as the number of amplicons increase, you get an increase in the amount of fluorescent signal in the reaction. To make a measurement from this, we take what's called a CT value. That's a measurement in time or in cycles of when the signal comes up above the background noise. Now I'll show you that threshold in just a moment. So here you're seeing some real-time data that's got two unknowns. Those are in red and green, and then some standard unknown or standard known controls. Those are in gray. So the threshold is when each of these curves crosses that threshold line, which it's supposed to be here. It's a horizontal line right around the value of 2 in this particular data set. Once we have that CT value, we then compare it to the others on this same run. So real-time PCR is a relative measurement because we need to compare those CTs to the others that are on this run. A CT all by itself isn't meaningful. It's only in context of the others that it takes on meaning. Now, digital PCR is different. It gets us an absolute quantity. So what you're seeing here is raw data from digital PCR. Each of these little dots represents a different subreaction from the same sample mixture. So this whole block that you see in front of you is from a single sample. It was divided out into 20,000 subreactions or microchambers. Every time the target molecule is present, that microchamber will light up. If it's not present, it'll be dark. So we count up the number of positives and the number of negatives in order to get to that absolute copy number of the target molecule or molecules. In the case of this one's a duplex in the reaction. To analyze this data, we create what's called a one-dimensional plot, and that's what you see in front of you now. This image is to digital PCR what amplification curves are to real-time PCR. All of the microchambers that are positive for the signal are plotted high up on the plot, high up on the vertical axis, and here you can see those in blue. And all the ones that are negative are lower. Those are a low level of fluorescence, so we count the negatives that are below that threshold, the positives that are above that threshold to get our absolute quantity. There's no comparison needed to any other sample. No standard curves, no reference. We can get that quantity straight from here. So when I think about how these two technologies compare with each other in the lab, I think of this graph, and I know it's complex, so let me describe it quickly. On the vertical axis, you see the amount of precision in the reaction, and it's being expressed in a confidence interval. So when you think about a confidence interval, you want that to be as tiny as you can possibly get. So the higher up on the vertical axis, the better. And then across the horizontal is the concentration of your target. And we're going from high concentration to low concentration. So with those axes in mind, let's start with the dashed line. So the dashed line that you see here is what's typical and theoretical for real-time PCR. So with real-time PCR, you have a huge dynamic range. You can see it's even going just like off the charts over on the right-hand side. You can put almost anything into real-time PCR and get an answer. And over the bulk of that range, you're going to have some really good precision. My graph almost makes it look like the precision isn't as good as it could be because it looks lower on the graph, but really the bulk of that line is really, really nice precision. And that's why real-time PCR is so popular in the lab. But for those of you who've already done real-time PCR, you probably have experienced what happens when you get out to CT values that are in the 30s. That's where your precision falls apart. If you had, for example, one molecule versus two molecules, that's a whole cycle difference. In real-time PCR, that would be poor precision. And that's why you see that dashed line just diving down into the horizontal axis. The precision just gets lost when we get out into those single digits of copies. Now, digital PCR is the asymmetric curve that you see over top of that. So it's got some significant differences from the real-time dashed line. So first off, there's some hard boundaries. If you look at the left-hand side in blue, that's what happens when every single one of those little microchambers is positive. If every single one of them is positive, then the system is saturated. It's overloaded. I can't determine a quantity from that. And so you see at that left-hand side in blue, the precision is down all the way to the bottom. I can't quantitate out there. And on the other side, on the right, where the red vertical line is, that's what happens when every single chamber is negative. There's nothing there. I can't quantify that either. But if I'm within those two boundaries, look what I can get for my precision. There's two really good reasons to use digital PCR. And the first is I could potentially access that peak of precision. And that occurs around 1 to 1.6 target molecules on average in the reaction. And just as a a little thing to give you an idea of how that looks. If about 1/3 of the reactions are negative, you would have about 1 copy on average per chamber. So I can see there that I have this peak that's so high, qPCR doesn't even come close to that. So that's one of the big benefits of digital, is to be able to get that really, really high statistical power. The other benefit is on that downslope going down towards the red. What you see there is, while it is getting lower and lower, it's still a lot higher than qPCR. So if you're looking to quantify something that's on the lower side, digital PCR can potentially give you more statistical power down there. So when I compare these two, digital PCR is a way to get an absolute quantity. Now, you just saw it has a narrower dynamic range, but within that range, it has some really excellent precision. I haven't talked about this yet, but digital PCR can potentially tolerate some efficiency problems. And the throughput for our digital PCR will range from low to fairly high, especially if you were to use the automated system that you just saw before I started talking. For real-time PCR, you can do a relative quantification. You can also do absolute quantification if you use an absolute standard curve. Real-time PCR has a really wide dynamic range. And over that range, it has very good precision, not necessarily as high as digital, but it's still good. It does generally require you to have uniform amplification efficiency. And the throughput for real-time PCR can go from low, but it can also go to extremely high with fully automated systems and 384 well plates. But let me dig a little bit deeper into that efficiency that I mentioned a moment ago. So what you're seeing here, this is some old data, and this is good data. You're looking at the amount of fluorescence in the log scale on the vertical versus the cycles. In the exponential phase of amplification here, you can see that those curves are what I refer to as, air quotes, generally parallel. That's what you want to see with real-time PCR. That means if you make some minor tweaks in the position of that horizontal threshold bar, if you move it up and down a little bit, the differences will remain the same because those lines are parallel. That's what you need to have with real-time PCR. But what sometimes happens, sometimes a curve might not be generally parallel to the rest. So this is an example of some bad data. If you look at that curve where I put the arrow, that curve is crossing over the rest. Now, I don't have a threshold positioned here, but imagine if anywhere in where the other ones are nice and parallel, I moved that curve up and down, that weird one would change in its difference relative to the others. That would be really bad news. That's why this is bad data, and you shouldn't use this type of data for real-time PCR. Now, with digital PCR, the only thing we ask in each of those little subreactions is just, is it positive or negative? So if this had been a digital PCR reaction chamber, it would have been perfectly fine because it's still positive. So that's what makes digital PCR so tolerant of inhibitors or efficiency problems. We just need enough efficiency to produce a positive when the target is present. So some of the other considerations as you're trying to decide between real-time and digital. Real-time has a great limit of detection. You could potentially put a lot of volume into a real-time reaction. You could do a 100 microliter one and boost your limit of detection a little bit. So limit of detection tends to favor real-time PCR. Limit of quantification, though, that favors digital PCR. Digital PCR has more statistical power at the low end to give you the better quantitation of low quantity targets. Some other things to be aware of. Digital PCR quantifies the number of molecules. So even though you'll hear all of us say copies, it's actually molecules. So if you were to have multiple amplicons on the same molecule that you wanted to quantify, you'd need to cut them apart in order to make sure that they are separate molecules. And then one other thing we're not going to cover today, but digital PCR is fantastic if you want to quantify single base changes or small insertions of deletions with high sensitivity. So let me just give you a few examples of how you might decide. So I'm going to start out with gene expression. For most gene expression studies, qPCR is a great tool. You should have enough statistical power to call twofold differences and higher amongst your samples. If you're using one of those QuantStudio real-time PCR systems that you saw in the opening of our session, those have even better statistical power. They should be able to get you to at least 1.5-fold. So that's great. You can use real-time data with delta-delta CT analysis tools or relative standard curves to do gene expression. Digital PCR will provide increased statistical power, especially if your expression differences could be less than twofold. Then you might want to go to digital. Another place where digital PCR can be a great tool is if you're playing around with those CT values that are out near 30 or higher. At that point, with real-time PCR, you often don't have the statistical power to call differences. That's where digital PCR can come in and give you that power to get answers at those really low quantity levels. Another benefit of digital is the answers are in absolute copy numbers, which makes comparisons really simple and straightforward. Another example of how I might decide, I was thinking what's going on in the world, and we've all been through COVID. So what if you were testing for that? Well, a lot of the tests out there have been done with real-time PCR. You can do a high volume PCR that'll give you great sensitivity. You'll have a huge dynamic range. You could go to high throughput using 384 well plates if you wanted to see if that target was present. And if you wanted to put in other targets for quality control, you can multiplex. But if you multiplex with real-time PCR, it takes very careful optimization and verification to make sure it's working properly. Now, digital PCR, remember I said, is very tolerant of inhibitors. So if you go out into the popular press, you'll see that a lot of municipalities are using digital PCR to test wastewater. So wastewater can tolerate those inhibitors, provided they're not too bad in that wastewater. It'll improve your limit of quantification if you're monitoring for changes in the amount. So if there's a certain baseline amount that's present and there's an increase, if you're talking about things that are very low amounts, digital PCR will give you the statistical power to call that there might be an increase or spiking in the amount of that target. Another benefit of digital is multiplexing is typically more straightforward with digital PCR, but it does require that all the targets be within that smaller dynamic range. Another great example of the comparison would be copy number variation. If you were doing a copy number variation experiment with germline material, where you might expect your number of copies of that segment to be somewhere between 0 and 4, real-time's a great choice. You would compare your samples to a reference gene and then a reference sample, get great quantitation. And if you wanted to boost your throughput, you could use 384 well plates. Now, digital PCR is where I would go if I was looking at, say, a somatic copy number variation. In those situations, the answer might not be an integer like it should be with a germline mutation. So digital PCR will give you the statistical power and precision to call that exact number of copies that are present in that material. It can also resolve higher copy numbers. So if you start getting out to more than, say, 4 or 5 copies of that target, digital PCR will resolve those higher numbers of copies. You can also run a single sample all by itself. You don't need to have a reference sample. But keep in mind, digital PCR measures molecules. So if those copies are in tandem, that would mean your amplicons are all in the same molecule, you would need to cut those apart for the best quantitation. So with that, I hope those give you some great tips in order to make your choice. Just as a summary, digital PCR takes your material, it divides it out into thousands of microchambers to do your quantitation. And then in each of those little chambers, all we're just asking is it there or not there, positive or negative? It does have a narrower dynamic range than real-time PCR, but the payoff is that you have access to higher precision and statistical power. It's also tolerant of inhibitors. Real-time PCR allows you to compare the timing of amplification among signals. Real-time PCR gives you a very, very large dynamic range, And while not as good as digital PCR for statistical power, it's still really good. And it's capable of the highest throughput level of PCR. And just finally, there's really not a right or wrong choice. Each of these technologies can be utilized in many of these experimental types. It's just which is the one that will give you the best outcome, the best data for your particular project. So with that, I'll wrap up and give you a few resources. So we have pages at thermofisher.com where you can learn more about the Absolute Q, digital PCR, and real-time PCR. I could point you to a web page, and that is thermofisher.com/absoluteq will take you straight to all of our digital PCR resources. And then finally, we have a podcast that's out there called Absolute Genius. It's a lot of fun, so I'd invite you all to check it out. So thanks, and I'll turn it back over to you.
Thank you, Marcia, for that engaging presentation. Now we're going to turn to our last talk of the day. Dr. Juliana Ramos is a senior technical specialist for digital PCR at Thermo Fisher Scientific. With a PhD in molecular biology from University College London, her scientific background includes roles in academia and biotech industries, researching new therapeutics against bacterial pathogens. Today, Juliana is going to be discussing some tips, tricks, and best practices to get you up and running with your digital PCR experiments. Juliana, I'll hand it over to you.
Today, we're going to talk about tips and tricks on the workflow of working with digital PCR, and in specific, of course, with the Absolute Q. So for today's agenda, we are looking at giving a brief overview of digital PCR and the technology that drives the Absolute Q forward. We are also going to talk about the MAP16 technology, so go a little bit more in depth onto the microfluidic array plate and how that works. And then we're going to mainly focus on the second half of this talk on workflow guidance. So we're going to go into details on sample preparation. This is going to cover what's important in that sample preparation to work with digital PCR, sample input, assays, how to transfer your qPCR assays to digital PCR, but also how to do an optimal design and some of those things. And then we're going to cover as well how to prepare a reaction mix and load on the MAP16. And those three main points on workflow guidance will ensure that you're going to have a good result at the end for your quantitation. I also added here the software 6.3 launch, which happened as of this week. So if you are currently already users of Absolute Q and are having the last version of the software installed on your computer prior to this, 6.2.1, I'm going to talk to you a little bit about the 6.3 version launch. If you have any questions, please feel free to write them down on the chat, and at the end of this presentation, I will make sure I try to answer some of them, and if not, I'll contact you individually. So just as an introduction of myself, I'm Juliana. I'm the technical specialist for the Absolute Q in the West Coast. There's 3 more colleagues working with me in North America, and I'm very happy to help answer any questions at the end of the presentation. So we start off with what is digital PCR. And digital PCR is going to be that technology that's going to give you a quantification of your nucleic acid target. And the main difference here, comparing to other technologies in the PCR world, is that you can do that without having a standard curve, without having a reference of known quantity that you can refer whatever is the CT value you get to get that number. So with digital PCR, we're working with absolute number of molecules per reaction. So in this particular slide here, what you're seeing are the 3 main technologies of PCR. So if we go back to that bulk PCR reaction, our conventional PCR, that gives us an endpoint read and only a semi-quantitative analysis of our target DNA. So what we can see here is that after our PCR is done, we can run an agarose gel way back when, and get an idea of band brightness, and that's as much as you can tell from those targets. Then if we go into qPCR, we started working with real-time measurements of amplification of our particular target. And we can do that by adding fluorescent molecules into our reaction mix. But that quantification that you're going to get on those amplifications are going to be relative to a known target. It's going to have to be relative and based off, analyzed through a standard curve. If we go into digital PCR, what you're going to get is an endpoint analysis, just like you would in your conventional PCR, but you're getting greater precision because you can actually count using fluorescent molecules as well here. So using primer probe set, for example, you're going to be able to get an absolute number and copies per microliter of that target. And the way we do that in dPCR is that instead of running that one well or one microcentrifuge tube, we actually are running over 20,000 smaller independent reactions from our bulk prep. So what happens here is that instead of running that one well, we are compartmentalizing, we are dividing our well into over 20,000 smaller independent reactions. And this is what our sort of overview of digital PCR will look like. So when we're prepping our digital PCR reaction, we're going to use the same chemistry as qPCR, because if you think about it, the technology itself is going to be doing an amplification of a DNA or RNA molecule. So we're going to use the same enzymes, the same set of primer probes, and a master mix for that particular application. Then we go into the compartmentalization or digitization, which is when you divide your sample into thousands of sub-reactions. And the idea here is that that compartmentalization is done in a random way, and you're going to have over 20,000 smaller reactions, PCR reactions, that are going to have enough reagent to amplify at least one of your molecules of interest. Then you go into the amplification, just like you're familiar with in PCR. And at the end, we're going to have a quantification by capturing fluorescence. And what you can see here in this example is a fluorescence of those over 20,000 reactions, smaller reactions, lighting up when you have your target of interest present. Once that is done, you can then count the number of positive and negative reactions and translate that number into a quantification in copies per microliter. And this is an example of what a digital PCR result will look like. What you're seeing here is a 1D plot. And basically what you see here is fluorescence per number of microchambers, those smaller compartments that you divided your sample in. And you basically can apply statistics and get a translation of this fluorescence acquiring into copies per microliter. So this is the sort of principle of digital PCR. You're going to divide your sample into thousands of smaller individual wells. If I ask you to pipette it by hand, a 30 microliter sample that has 3 copies of your DNA in 3 individual tubes, what's the probability that you're going to have 1 copy per tube? That's a random distribution, right? The actual probability is quite high, but you can end up in digital PCR in a scenario where you're going to have more than 1 copy of your target within each well. In digital PCR, we cannot distinguish between that number of copies. We can only distinguish between positive and negative wells. That's sort of the technology today. So how do we actually correct for those molecules, those chambers containing more than 1 copy of your target? Today, the technology uses Poisson statistics to be able to correct for reactions containing multiple molecules. Provides you a probability of how many molecules you would have within that sort of distribution area that you divided your sample with. So as long as you have 1 negative reaction, you can apply Poisson statistics and get that correction factor in your compartmentalization. So this is a scenario that happens here where you have those positive and negative microchambers that are with the presence of your target or with the absence of your target. It's called digital because it's based on a binary system. So your answer is always going to be 1 or 0, positive for your target or negative for your target, or full fluorescence. Here, how you digitize, how you actually do that compartmentalization is going to be what determines the quality of your digital PCR results. And that first step of digital PCR, if you think it as a technology that has 3 main steps: compartmentalization, the PCR, and then the counting of those fluorescent molecules. Basically, what you would see here is the main difference between multiple technologies in the market today is how you do that step 1, that compartmentalization. Some of the benefits of digital PCR is when you actually need to get that absolute quantification, that number, without a standard curve, either because you don't have a particular reference gene, or you know how standard curves can be cumbersome sometimes to be able to perform and get an accurate value. What you're going to get in terms of results is what you can see here in this table on the left, an average copy per reaction, and you can then basically compare between samples your absolute quantities. So your analysis of your results are going to be very straightforward. Digital PCR can be a really good technology when you're also looking to analyze targets that are low in abundance. If you compare with qPCR, we are pushing down on the concentration scale that we can detect in digital PCR, and we're gaining precision to be able to do that. Digital PCR is a technology that requires you to work with a smaller dynamic range, exactly so you don't saturate all those microchambers with positive wells, but it's really good to analyze those targets low in abundance. Here, you're not going to end up having competition between, let's say, wild type versus mutant allele. That can happen in qPCR. When you want to monitor small changes over time, so let's say you found a biomarker of interest and you want to, over time, sort of monitor for treatment efficacy, for example, you can use this technology to be able to discriminate very low fold changes between treated and untreated samples, for example. And when you need a solution that is somewhat resistant to PCR inhibitors, imagine that you have a particular sample that has high inhibition, high PCR inhibitors, high content of PCR inhibitors. Once you are dividing or compartmentalizing that sample, it's almost like you're adding somewhat of a dilution factor to your potential inhibitors within that sample. So this is the overview of the technology of digital PCR. And the Apply Biosystem QuantStudio Absolute Q dPCR system works with a microfluidic plate format to do that compartmentalization. And this is what the Absolute Q looks like if you're not already a user of the Absolute Q. So it's one single instrument that allows you to do those three main steps: compartmentalization, PCR, and scan of your results in one single box. Basically, here are a few features of the Absolute Q that I usually like to sort of remind users that are improving your data. So the first one is reagent efficiency. So with the MAP16, which I'm going to introduce to you in a minute, how we do our compartmentalization, we managed to increase reagent efficiency. So today we have less than 5% of wasted reagents during that first step of division of your sample. So here we are working with a particular array that doesn't have an input and output channel. It's sort of whatever you add your sample in is going to be staying within those compartments. You don't have that volume in here. And you have a consistent 20,000 microreactions that you can analyze. So really what passes the QC of the equipment is always going to be over 20,000. Those two main points are going to directly impact on your Poisson statistics, that statistics that translates your positive and negative fluorescence into a number that you can then do your analysis. In terms of workflow, is very straightforward and easy to use, equivalent to what you do today in qPCR. And our standard runtime is about 90 minutes. We are today one of the only equipments that can do an autophos positive rejection. Basically, our instrument doesn't have any robotics inside. It allows us to take what we call a pre-scan photo and subtract any potential false positive background fluorescence. It's a single instrument, like I mentioned. It requires very minimal maintenance, and you can have a flexibility to work with low throughput all the way to high throughput. Our instrument, the Absolute Q today, is compatible to do 4-color multiplexing. And this is what the workflow of the Absolute Q looks like. So like I mentioned, step 1, you're going to prep your reaction. Step 2, you're going to load it in a plate, which is a technology that drives the system. And then you're going to run your PCR and collect your data. You can analyze your data in our Absolute Q software, which we're going to touch base in a little bit. And this is what the MAP16 technology looks like. It's a microfluidic array plate that has 20,480 fixed microchambers per array. Those microchambers have a fixed volume. Everything you add on it will get analyzed. So that really accounts for the very low data volume that we have. And here what we can see is that 99% microchamber field success rate. In this particular graph here that you can see, you're seeing the comparison of a MAP16. So this is a comparison of a dilution series or a replicates compared with the droplet-based system. And here what you can see is the total number of microchambers that were accepted per reaction. It is within the MIC guidelines that for you to have the highest precision in digital PCR results, you should be accounting for over 20,000 positive reactions that you can read. By positive, I mean passes QC of the equipment are being able to be analyzed. So this is a really good consistent result and success rate that we have with the MAP16 compared to other systems. And the second point that I mentioned, and I mentioned that it will impact on your Poisson statistics and your quantitation at the end, is the dead volume. So here again is comparing the MAP16 with the droplet-based system and 5 dilution points, and just showing that wasted sample per reaction with the MAP16 is very, very minimal compared to the droplet-based system. And the way the MAP16 works is more or less like this. What you can see here in this first figure is the MAP16 with 16 samples, 16 wells that you can run. And those wells, they can run in intervals of 4. So let's say in a particular day, you don't have a lot of samples to run. You can run the MAP16 in intervals of 4 and save that consumable to use later on. The way our compartmentalization is done in the MAP16 is by applying pressure through those gaskets that you can see here. So those gaskets are how you close your wells in the MAP16, and they have that hole in there where air pressure gets applied and basically pushes your reaction mix from left to right through all those smaller microchambers. Each microchamber here has a fixed volume of 432 picoliters, and that is consistent. Once you apply that pressure and your reaction mix gets filled into all those microchambers, what goes right after it is an isolation buffer that sits in between those canals and makes sure that each individual reaction is separated. So there are individual PCR reactions. Imagine this as being like an ice cube tray. You know, you're going to fill your reaction mix all the way to the top, and within those lines is going to sit that isolation buffer. That's the technology of the MAP16. There's no sealing here. There's no complex sort of technology. It's basics in microfluidics. What you can see in the plate, and this photo doesn't do justice, but it's a plastic plate with a semi-permeable plastic bottom. So it's very robust against air bubbles that you might insert or pre-existing air that could be in microchambers. And this is a snapshot of what the results can look like in digital PCR using the Absolute Q software. So what you see here on the top is the MAP16 diagram. And basically right next to it, you're going to see your table of results. And basically in digital PCR, your table of results are going to consist in your concentration in copies per microliter. So the software already does that Poisson statistics for you and gives that data already calculated. It's going to give you a 95% confidence interval, so your upper and lower confidence interval within that number on that distribution. The total number of positives, so all those reactions that emitted fluorescence within those 20,000, and the number of total microchambers that passed QC. So again, over 20,000 reactions. So here what you can see is that in a total of 20,480, what you can see here in this slide is what passed QC is around 20,350. And then below here, you see a few ways that you can see that data in digital PCR. So that 1D scatter plot graph that I showed you in a synthetic image of what that sample being divided into those 20,000 arrays look like. And basically in the 1D scatter plot, all that's happening here is that the system is clustering in 2 different clouds, positive reactions that emitted fluorescence versus negative reaction, and setting somewhat of a threshold that we call here, different from the qPCR threshold, between those 2 clusters. And on the next here, if you're doing multiplexing, you can also see your samples as a 2D scatter plot. What you can see here as the QC image on the top right on this slide is that basically we use rocks as our reference dye here that can evaluate both the distribution of your sample within the array, but also the fluorescence level between each microchamber. And it's nice that you get a photo of that ROX QC so you can see how well your loading is in each individual microchamber. So if everything is done right, you should have ROX emitting fluorescence in all the 20,480 microchambers. So this is a scenario working with a MAP16 using 16 samples and then low throughput analysis. If you are needing high throughput to run with the Absolute Q, we are now working with the Auto Run suite. And basically that allows you to attach in your Absolute Qs a robotic arm, the spinning curve microplate mover robot. And basically you can stack your plates into hotels or stacks. And basically the robotic arm will be loading your plate in and out for you of that instrument. You can connect it to one instrument or a couple instruments and gives you a true walk-away automation. Different from having multiple plate drawers in one instrument, having that arm outside of the instrument really allows you to not have to wait for your run to be over to then remove those other plates and put new ones. You can stack about 80 plates in that hotel and have a true walk-away automation. A nice thing about the robotic arm is that allows us to not change the QCs already present in the Absolute Q. So if you remember, I mentioned we have that pre-scan because our plate stays in stationary in our Absolute Q. So we can take that pre-scan photo and subtract any background fluorescence to give a much higher confidence, especially if you're working to look for those rare mutation targets. And allows us to do batch analysis. So let's say you have a 96 well experiment or a 384 experiment that will require you to pipette a few MAP16s, which will give you the number of samples you need to analyze, and you just tell the system that this is one study. There's very minimal plate-to-plate variability, and again, no variability within your wells in a MAP16, so you can get a very tight CV on those data points as well. So this is how you can automate if needed. So this was a brief overview of our Absolute Q and of digital PCR as a technology. And now we're going to go into the main topic of this presentation today, which are tips and tricks on the Absolute Q workflow. And if you already work with the Absolute Q, you can write it down in the chat and we can have a deeper conversation as well if you want. But I'm going to cover some topics and I'm going to start with sample preparation. And again, this is true for any digital PCR, but when we go into sample preparation, I usually get asked a few questions. So what samples can you run in dPCR? You can run in digital PCR any extracted genetic material, so DNA or RNA, that can come from multiple biological sample origins, so environmental DNA, tissue, cell culture, blood, bone, etc. Can I run samples without having to do an extraction or purification? So we have tested in the Absolute Q several examples of crude lysates from bacterial cell cultures, mammal cell cultures, crude lysates from plant tissues and others. And the answer is yes, you can run. It will require lab validation, but it can be run on the Absolute Q. And here's just an example of one of our application notes working with AAV particles. So what you can see here in this graph is 5 different plates for an AAV particle, 5 plates for that sample type, and comparing it to an AAV control DNA. And here, and on the y-axis, you have the concentration in copies per microliter. So on those AAV particles, all that was done with that particular sample was a heating step, basically, a 95-degree for 5 minutes heating step to break open your viral capsids. No extraction or purification here was really done in this particular sample type. And you can see that all the samples fall within 20% overall mean concentration compared to the control DNA. And again, here's just showing that plate-to-plate variation is not significant. So we're seeing a CV percentage of less than 3.2% in all those experiments. So what are the main things to consider when we're talking about sample preparation in digital PCR? A few things to consider are sample purity, your sequence and target length, your genetic material integrity, and sample viscosity. We're going to touch on a few of those today, and I'm going to start with sample purity. Even though I did mention that digital PCR can be somewhat robust against inhibitors, sample purity will be a factor that's going to impact any PCR reaction. here, right? This is an example of testing different RNA targets. So here's 3 RNA targets with our one-step RNA master mix in the absolute Q in the presence of common PCR inhibitors. So here we have 4 common PCR inhibitors. And what you see as that mean concentration is coming from a no inhibitor control sample. And you can see that those concentrations, they will remain about 30% within that 30% of comparing to the control sample. So it is somewhat robust against inhibitors, but inhibitors can impact your reaction if you know you're working with samples highly concentrated in inhibitors. They can reduce your fluorescence, they can sort of reduce your resolution, giving you less separation between those clusters of positives and negatives. And they can denature your tight polymerase, right? So if you're working with high amounts of alcohols, salts, nucleases, and others, that can definitely have an impact on your PCR reaction. So a few tips that we can give here is that if you already know you're working with this particular sample type, and an example that comes to mind is when you're working with environmental DNA, usually those samples will have a higher amount of inhibitors. You can basically dilute your samples further in digital PCR to sort of achieve a nicer resolution in your data. And you can always run that positive control to check for your reaction efficiency. Again, I like to mention this, but also remind you that we are doing an endpoint measurement here in dPCR, and that's why it can be robust against those inhibitors. So your endpoint measurement is not going to be affected by the efficiency of amplification in each cycle. A second point I'm going to make here as well is when to add a restriction digestion step when prepping your sample. So it is known that when you're digesting the DNA that it's, of course, outside your amplicon region, you can reduce digital PCR range by increasing that accessibility to your target for your parameters and probes. That is a given for other systems that you have to do restriction digestion in your samples to reduce that dPCR range. When you're working with the MAP16 with those fixed microfluidic wells, this is an optional step. So usually when you think about samples that will look better in PCR when you digest, plasmid DNA with all those secondary structures come to mind. In the absolute Q, it's not a required step. So you've tested it several times and plasmid DNA does not necessarily require that linearization. Other sample types that could require that in other platforms are samples with high viscosity or high DNA concentration. With the MAP16, as long as you can maybe do a further dilution step for that particular sample, and with high viscous samples, another thing that I usually recommend is that you make sure you vortex really well that sample, maybe heat it a little bit to reduce that viscosity. That is probably enough. So the sample prep is more important than necessarily having to do a restriction digestion step with those particular sample types. When we see improvement with restriction digestion is when we're working with whole genomic DNA, especially from plasma and blood samples, and when we're doing CNV assays, when we're looking for those linked tandem sequences. And those CNVs will require digestion just so we can ensure the accurate analysis of the individual target genetic region and get a true copy number count. So this is an example of a data in the absolute Q with no digestion and with digestion with a CNV assay. So it's comparing a CNV assay, and what you can see here for this particular assay is with no digestion, we get no separation between clusters. And with only 1 unit of this particular restriction enzyme, we already get a much nicer cluster separation here. So what are the to-dos here when you are assessing whether to do a restriction digestion step? What does it sort of look like in our workflow? Basically, you don't need to do any pre-incubation. All we recommend here is that we even have a protocol that we can send your way for a 1-pot restriction digestion step. Basically, that means that you would only require to add into your reaction mix whatever it is the restriction enzyme you're using, and you don't need to have pre-incubation time. You can just put that reaction mix right into your MAP16 plate and put it in the instrument. If you want, you can leave it at room temperature for, let's say, 10 minutes, but it is not required. So all you would do is add your restriction enzyme within your reaction mix and load it in the MAP16. In our system, different from most of other systems that require a large amount of restriction enzymes, 1 unit for TechMin assays is more than enough. So a few tips as well here that we've seen on the field is that if you want to use more than one restriction enzymes, you can. That also works. And if you're working with TechMan pre-designed assays, you can request to your tech support the padded ampli-consistent sequences in between, sort of outside your target region, to find what is the best restriction enzyme that you can use for this particular example. Question I usually get is sample input. So how much DNA or RNA material can I work with? So sample input amount will depend on the digital PCR technology you're using and how that compartmentalization is done. So here for the absolute Q, the dynamic range of the instrument can detect from 0.1 copies per microliter to 10 to 15,000 copies per microliter within that reaction mix. So when you're working with DNA and your target is known, for you to be able to determine that optimal dilution to start off in the digital PCR, you got to determine the copy number of that target per genome. So once you find that copy number, you can then dilute your extracted gDNA to what will be the ideal input range for digital PCR. And here we give that range of 0.6 to 1.6 copies per chamber. And I can introduce you again to the idea of lambda in the Poisson statistics in a minute. And that will be sort of the sweet spot of digital PCR. A few quick tips here is that you can use our DNA calculator to check for that genome size. And there's also on the bottom a database that will give you that copies per genome of some genomic variants. So why 0.6 to 1.6 copies per chamber? So when you go into Poisson statistics and you're looking for that precision in your absolute quantification, that precision is going to be dependent on your sample concentration and the total number of microchambers you're analyzing, right? So here we have in this graph your percent deviation, percent of PCR reactions which are negative, and your copies per reaction on top here, which is lambda, basically, in the Poisson statistics. What you can see here is that you're going to get the best precision in dPCR, so a much shorter percentage deviation on your upper and lower 95% confidence interval when you are at a 0.6. So if you can see my cursor here, 0.6 to 1.6 is where this data here is tighter. So of course we are aiming to have that one copy per reaction, but as you can see in this graph, from 0.6 to 1.6, that percentage deviation on your 95% confidence interval is tight and is within digital range. So here's an example using RNA speeding, just so you can understand how you can do that calculation. So basically, you're going to have to find the ratio of genomic copy per mass of the genome that you're analyzing. So for RNA's P gene, you have 1 copy per 3.5 picograms of human genome mass. And again, you can use those tools that I put it here in the slide to be able to calculate that. So you can infer that 1 copy of that target will be found in that amount of human DNA. For you to find that sample dilution to be within dynamic range of digital PCR, you're going to get that target copy number and you're going to divide by the volume of your microchamber, which in the MAP16 is always the same. So we're working with fixed microarrays, 432 picoliters. Once you do that calculation, you can then know that you have to dilute your DNA stock to 8.1 in this particular example, nanograms per microliter. Then you are going to expect to see 1 copy gene target per reaction well. Of course, samples should not be diluted when you already know your target of interest is rare or low in abundance. And here is, if that target is potentially highly concentration, you will have to dilute to avoid reaction saturation. A different scenario is when you're working with a target that is unknown. So in this example here, we're talking about, that locus of unknown copies per genome or RNA. You're working with that mRNA with unknown expression levels. What are you looking at? Usually the step here will be to prepare a dilution series of that sample, approximately 4 data points, so you can be above and below the digital PCR range. There is a recommended concentration of RNA per dPCR reaction, so 1 to 4,000 copies per microliter per reaction. But if you have no idea of how much you're starting with, using qPCR can be a good sort of tip for you to be able to estimate the target molecule input. So you can even use your real-time PCR to estimate the target input for that dilution series prior to go into the dPCR. You can kind of infer that you have one copy in, let's say, 20 microliters, will produce a CQ of 38 in qPCR. And you can use a CT to copies guesstimate table that we call. And ideally, you're going to have one copy around the threshold of 35 to 40. And this is just an example of a guesstimate table that you can make. And if you have a qPCR in your lab and you don't want to work with dilution series of each particular samples, you can use that or historical CT values to have a guesstimate. So controls. Which control should I run in my digital PCR experiment? So definitely a positive control and a negative control are needed. So to get, that template amplification control under the conditions you're going to be working with in the Absolute Q, and the negative control to monitor for assay efficiency and to determine that limit of detection or that limit of sort of the threshold that you set between those clusters. Non-template controls for control of that contamination in reagent and known assay control are also options for controls that you can run within your digital PCR. And then I always get asked, do I need to run endogenous controls if I'm working with mRNA for having that control of extraction or input, depending on what you're working with? If you want to use an endogenous control to have that extra sort of analysis, you can. Just remember, in digital PCR, you're going to be counting total number of molecules per reaction to get an absolute quantification. So your endogenous control is not going to be doing the same thing as it will be doing in qPCR. If you are using an endogenous control, you would have to choose a gene that has a similar expression range of your particular target, so you don't saturate or have to do then serial dilutions for your endogenous control and add a lot of back calculation steps to get within the same dilution range. We have with Thermo Fisher a sort of comprehensive panel of endogenous controls for qPCR that you can run together with a sample to find what will be a good endogenous control if you're interested in running that with dPCR as well. The next point here is digital PCR replicates. So do I need to run replicates on my digital PCR experiment? So when you think about digital PCR, that first step is actually determining or sort of compartmentalizing your sample into over 20,000 reactions. Is almost like it's adding 20,000 technical replicates of your one reaction. So there's no need for technical replicates in digital PCR once an assay is validated. Like I mentioned, we have no well-to-well variability and very small plate-to-plate variability. So once your assay is validated with the correct controls, positive and negative controls, you don't need to have to do technical replicates. And what you can see here in this graph is an example of a number of a sample replicate of an N of 15 on the map 16 in the Absolute Q. And you can see those very small error bars within each sample and a CD percentage for this particular target here of less than 3 here, right? 2.4. So giving you a very, very tight data. This is a particular example where we actually ran this is a 4-plex assay. And for all the dyes, all the targets that we're looking at here, we got a CV% of less than 3% between 15 replicates in the map 16. Biological replicants, of course, are recommended based on your experimental design and your biological question. And I think one point even before I go here into prepping your samples is that when you are reporting your values of digital PCR, the MIC guidelines, sort of the checklist, considers essential that you publish, when you're publishing your dPCR result, some of the informations, and one of them is experimental variance. So they prefer that you run multiple biological replicates, and if you're working with a system that you can show that you don't have well-to-well variability, you can certainly not need to have to do those technical replicates as well, which is the case for the map 16. So preparing your samples to load on that array. So to get successful filling of all those 20,000 microchambers here, sample vortex and centrifugation are key. And it might seem simple here, but we're not talking about that quick vortex of your sample. We're talking about a 30-second vortexing of that sample after extraction and centrifugation. Once that sample is extracted, vortex and centrifuged, then you can go and add into your reaction mix, into your super mix with your enzymes assay to then load into the MAP16. This is going to reduce the risk of carrying any potential large physical debris over to your reaction. The MAP16 itself is incredibly robust against any obstructions. We are working with a microchamber that has about 30 microns in size. So here, when we're talking about physical debris, we're talking about large particles. So let's say large soil or dust within the environment, not any potential other smaller debris or cell lysates or things like that. Because as we mentioned, we can work with crude lysates here. So here is just to avoid any potential large debris. So what you see here in the photo is a syntactic image that represents correct filling of the MAP16 over 20,000 reactions. And this is an image of what you can see a microchamber being obstructed due to large particles present within those samples. This is what it would look like. So once that is done, then you can prepare your reaction mix. And once that reaction mix is ready to go, you should make sure that you also vortex that and centrifuge well to ensure distribution of that ROX, which is our QC dye in our master mix, to be evenly distributed across all the microchambers. Again, here we recommend to do a little bit of an overage to pipette it from the top of that reaction mix, and again, avoid the carryover of any large particular debris. A quick tip here in digital PCR for any system is that volume matters. Having consistent volumes and pipetting volumes within each compartment will really determine success on your quantification. So working with properly calibrated pipettes are needed. Here is just an example of what a proper reagent distribution looks at your QC. So a CV% of 7.4 and a QC image where you see ROX that's present in our master mix evenly distributed in all the microchambers, showing correct filling, no obstruction. And here is an example of what that will look like if you don't have equal reagent distribution. So that's why that vortexing properly once your reaction mix is ready is very important. Then we go into loading the MAP16 with the reaction mix. And here I'm just sort of showing you a sort of protocol for one reaction in the Absolute Q. And I'm just sort of pointing out that we have a master mix here that is a 5x master mix. So more concentrated than you're used to in qPCR, allowing for you to play around with how much sample you can add within that reaction mix. So your reaction mix is not going to be prepared in the MAP16 at all. It's going to be prepped either in a 96 well plate or a microcentrifuge tube. The MAP16 does not get vortexed or centrifuged. Those arrays right next to your well have to maintain itself clean. So when you're loading in the MAP16, you can just place the MAP16 in a clean surface. And you do not need to use air duster or anything like that to clean the MAP16. We actually recommend you not to. Then we go into our digital PCR assays. So if you're working already with qPCR assays, almost any Techman assay can be used and transferred to the Absolute Q. I'm going to use the same primer probe concentration within the reaction. So 900 nanomolars for forward and reverse primers and 250 for your probes. If your assays are already correctly designed with a nice melting temperature, amplicon length, and based on sort of all the aspects that you need to think of when designing an assay, and you still find a scenario where you can see here in this photo of digital PCR rain on the example B here, and that digital PCR rain is comparing to the sample A where you get nice resolution and division of the clusters, there's a few things we can start playing around with sample analysis and troubleshooting. That can be taken into account here, and they most likely will be protocol changes. So if you're working with, let's say, GC-rich sequences, you can increase your denaturing time. So you can then play with your digital PCR protocol. But again, most of this digital PCR rain can definitely be already assessed in those pre-steps, in those sample preparation steps, whether you need to use a restriction digestion or not. If you're still getting, after properly designing your assay, and specifically if you're working with pre-designed assays that you should be just plug and play, then there are definitely a few other tips and tricks that we can investigate on your particular assay. So let's say if you're finding non-specificity as well, you can change the temperature and time of your annealing and extension protocol. So what you're expecting here in digital PCR is to get a result that looks like photo A, with very nice resolution and division between positive and negatives. And for the Absolute Q, we have several pre-designed assays already. So we have assays for a range of applications, from liquid biopsy to environmental DNA, CNV assays. But we also have, if you're looking for doing custom multiplex assay, the Absolute Q tool for designing that assay. And what you can see here is that there's 3 main steps. It's very straightforward. You can choose your application on the website. Then you basically input your target sequences. You're going to review your design and check out. If you need further complex modifications or multiplexing, that's where you contact either me or your tech support or field application scientists to get support on doing those designs for the Absolute Q multiplex assays. And then they go through extensive bioinformatic and silico analysis, so you can get the best option for you to start with your multiplex. This is sort of the overview I wanted to give on the workflow. And then I just wanted to touch base on the software launch that we just had, software 6.3. So if you're already familiar with the Absolute Q software 6.2.1, a few changes you're going to be very excited to see is that we proved the efficiency in analysis and sort of streamlined run setup templates. So you can just upload your Excel template and you would have a much faster setup of your run. And for me, one of the best changes here is that the analysis is about 9 times faster. So for you to populate all those graphs, if you're working with large groups of samples, that's going to be extremely simple, and it's going to be a click of a button rather than having to wait for those graphs to populate. We have a better understanding of our QCs. So you're going to have QC flags for each sample, understanding whether that was a problem with distribution or if you have a sample that particularly failed, you're going to have a lot of QC flags to explain better what is going on within that sample. You're going to be able to do batch creation. So having those studies up to 384 samples, like I mentioned, if you're going to high throughput, and even if you're not having an auto run a robotic arm within, you can still do those large throughput experiments. And I didn't mention earlier, but the MAP16 is compatible with Liquid Handler. Several Liquid Handlers already have protocols for it. And here you're going to have enhanced analysis. So you can set thresholds in advance, let's say, amongst several plates and batches, and you can have a better interpretation of your data. The results are going to give you more data points, and you can actually integrate your dilution factor calculations already within the setup of your run. So let me just quickly show you what that will look like. So first here is how you set up your plate. As you can see, you already have the dilution factor option in there that you can place what is the dilution factor of that particular sample, and it can already back calculate for you when it's giving you that copies per microliter result. You can save and set up sort of those templates that is going to be much easier for you to be able to use it. And another nice feature here is that you can set your threshold prior to the run if you're working with an already validated assay that you want to make sure that each target channel is always being analyzed in the same threshold. So that was what I wanted to cover today. I'm going to leave you here with a few slides that have some of our most recent publications leveraging the Absolute Q for different applications. So cancer research, cell and gene therapy, environmental surveillance, and pathogen detection. And if you want to take a photo of those QR codes, you can. I can also send some of those publications to you if you're interested in going deeper on them. And also in other applications here as well, some review articles, transplantation research, and others. So I just wanted to thank you for your attention and for being here at this webinar today. This is my email if you want to email me directly with any questions. We will have potentially a date yet to be deep, a dPCR Academy going on to the analysis of your data then within the new software, so you can understand a little bit better how to analyze your digital PCR results in the Absolute Q. I don't think I can see any other questions. Please feel free, like I said, to email me with any questions, or if you want further information on the Absolute Q, I'm happy to have a one-on-one meeting and discuss.
Thank you, Juliana, for that informative presentation. And as a reminder, all questions that were asked during this section will be addressed post-event. And finally, I want to thank all of our attendees for joining us at For the Love of Digital PCR 2024. We hope you found these sessions informative and inspiring as we explored the remarkable capabilities of digital PCR. Remember, this virtual event is not just a one-time experience. It's a year-long journey filled with ongoing content updates and insights from invited guests. We encourage you to continue coming back to our virtual environment where you can access a variety of downloadable content. You can also request more information from our sales representatives at any time through the Contact a Specialist forms that you see throughout the event. Thank you once again for your participation, and we look forward to seeing you back at For the Love of Digital PCR 2024 for more exciting content and opportunities to connect with the digital PCR community. Keep exploring, keep learning, and keep pushing the boundaries of scientific innovation.